Hello, this is Eva Amson welcoming you to this Bite-Size Bio webinar, which today is sponsored by TomoCube. TomoCube provides holotomography imaging platforms for label-free 3D quantitative imaging. HTX-1 holotomography makes it possible to analyze subcellular details of live cells and multicellular specimens such as organoids or tissues without any modification from pre-treatment like fixation or molecular labeling. The quantitative information can be easily analyzed with the Tomo Analysis software. Today's presentation is titled Label-Free 3D Live Cell Imaging and Quantification Using Holotomography, and it's being presented by Yonghyun or Paul Park, CEO of TomoCube. Paul is an endowed chair professor of physics at KAIST. He earned a PhD in medical science and medical engineering from Harvard MIT Health Science and Technology. His area of research is optics, holography, and biophysics. He is a fellow of the Optical Society of America and the Society of Photo-Optical Instrumentation Engineers. He received medals of honor in science and technology from the President of South Korea and the Jinky Hong Creative Award. His research has led to the creation of 2 startup companies with over 80 employees, The Wave Talk and TomoCube. As always, we will have a question and answer session after the presentation, so please type any questions you have into the questions box, which appears on the top right panel of your screen, and I'll put them to Paul at the end. Details of how to access the on-demand recording of this webinar will be sent to you by email shortly. So now over to you, Paul, for the presentation.
Welcome to join this webinar. My name is Paul, and I'll be talking about the concept of holotomography, and I'll be share what you can do using holotomography for the study of the live cells and tissues, use it without using any labels, and provides the quantitative analysis. To make a long story short, you can visualize the complex shape and dynamics of live cell without using any fixations or staining or any preparation. Now you're watching cancer cell line, and you can see individual subcellular organelles such as nucleus and nucleoli. And now you're watching mitosis. So you saw the condensations of chromosome and like vivid motions of the lipid droplet and cell membrane. So this is how holotomography can visualize in order to study the cells, the physiological states. And for now, I'd like to talk about the principle of the holotomography system. And I'd like to talk about the history of the live cell imaging and what are the major milestones in the history of the live cell imaging and the relevant microscopic techniques. Ernst Abbe and Carl Zeiss has provided the foundations of the modern optical microscopy, bright field microscopy system. So they have defined the resolutions and provided the high quality objective lens so that you can have the you can imagine imaging optics by magnifying the small object to the large size. But the problem is the cells are very transparent because they are mostly made of water and proteins. Without any staining, they look very transparent, almost not invisible. In order to solve these issues, the Fritz Janicki, Dutch physicist, who came up with a very clever idea of using phase contrast. So by modulating illumination and modifying the objective lens, you can convert such a low contrast imaging of live cell into high contrast imaging, such that you can see the cell boundaries or the nucleus presence. But this is 2D and qualitative. And during 1990s, scientists has developed a series of techniques to fluorescently label specific proteins using fluorescent proteins technique, so that which has opened the doors to molecular biologists by adapting the fluorescent microscopic technique. So now you have the great molecular specificity. So now you see that where is a specific proteins are located in live cell. So that has opened the door to the molecular biologists, but still you have to worry about the effect of the staining or the introducing the fluorescent proteins. the genetic modification and the photobleaching and phototoxicity are the major bottleneck. And more important thing is those modified cells cannot be used for the therapeutic application. Now we are witnessing the errors of the 3D biologies and regenerative medicine. The model cells samples are becoming more and more complicated, such as the organoids and spheroid and Lebanon chip. And now you have to look at the multi-layer and the multi-structured the cellular systems with high resolution. And stem cells and organoids need to be used for therapeutic applications. So the use of the fluorescence is not allowed, and it is inevitable that you have to use the label-free technique. This is why we are working on the label-free 3D and quantitative imaging technique. And let me briefly talk about the challenges in 2D and 3D imaging of live cell. In 2D, as I said, the most powerful driving force nowadays is fluorescence-based microscopic techniques, such as confocal microscope or light sheet or those kind of the super resolution technique. But they are all based on the use of fluorescent protein. So you have to spend some time to prepare the cell in order to present the specific fluorescent protein. It takes like hours and days. And even though you may get very high contrast imagings with the powerful molecular specificity, those fluorescence-based imagings are not highly reproducible because level of fluorescence are varying depending on cell types, and they are not very quantitative. So the analysis of those cell imagings should be very subjective and not easily automated. Compared to this 2D imaging contrast, when you use the holotomography system, you don't have to worry about the preparation, no staining and no fluorescent protein expression and no other protocols. So once you have samples, you can easily and rapidly visualize the cell. And what you utilize is the refractive index distribution, so which is highly reproducible data because this is the physical quantity. And you can also retrieve various quantitative imaging parameters, so such as protein concentration and volume and dry mass of the subcellular organelles, such as cells, can be quantitatively calculated. And furthermore, you can also perform the long-term imaging because we do not worry about phototoxicity and the modification of cell. You can measure cells behavior for days and weeks compared to conventional methods only measure like up to few hours limitation. And now we see the 3D complicated the biologies. So in order to visualize the samples such as organoids, conventional technique requires fixation and sectioning, sometimes like staining, which means you basically kill the organoids or the highly complex 3D architecture of the cells and tissues. And so you measure a seed shot of that organoids, and furthermore, that also not be able to be used for therapeutic application. Whereas the holotomography provides the label-free imaging technique so that you can keep watching the growing the organoids over a long period of time. And 3D structures can easily measured up to some size up to 150 micrometer. And since we haven't modified anything to these organoids, you can use those organoids or the iPSC for therapeutic application because there's no light energy transfer and no staining, no modification. So you can just keep those samples and the model system alive and in their intrinsic stage. The principle of the HET is the optical analog to X-ray computed tomography system, X-ray CT. In CT, it measures multiple 2D X-ray images, projection images as a function of various illumination angle, from which you can reconstruct three-dimensional distributions of X-ray observability so that it can provide non-invasive tomographic visualizations of the internal organs we can do the same thing with light. So by measuring multiple 2D hologram, and from which you can retrieve three-dimensional distributions of the refractive index, and then you can visualize cell and subcellular organelles without any preparation or any staining. So even though those two techniques look different, they share the same physical principle. But in HET, we use light and we look inside of cell, but everything is same. So using HET, you can visualize the live cell, unlabeled cell in 3D in quantitative numbers. So if you look at the right-hand side, you can see the still shot of the live cancer cell line. You can see cell membrane and nucleus membrane, nucleoli, mitochondria, and those whitish spheres are corresponding to the lipid droplet. Whereas if you compare conventional the label-free techniques such as phase contrast or DIC, even though they do not require labeling, but the resolution is sometimes very limited to low resolution, and it only provides 2D and qualitative, so widely used for the cell counting, but not used for studying the detailed structures of the live cell. If you use fluorescence, it can provide the strong molecular specificity, but you have to worry about phototoxicity and photobleaching issues, as I mentioned. And also the number of the labeling dyes are limited because of the experimental complexity, and as well as the light efficiency becomes low if you have more than like 3 to 4 fluorophores. Since we can also measure multiple 3D tomogram for a certain period of time, you can see intriguing dynamics. So now you are watching here the mitochondrial fission and fusions, and you can also see that cell chromosome condensations during mitosis. So those three-dimensional structures can be easily observed and quantitatively analyzed. This is examples of showing the same cells using different modality. And if you use bright field microscope, since unlabeled live cells are very transparent, you almost see nothing. But if you use fluorescence, in this case, we have fluorescently labeled mitochondria. You can see the presence of mitochondria, and you can see the locations of mitochondria. But the other part of the cell structures are not visible in this case. But if you use HET, you can see overall cell shape as well as the subcellular, major subcellular structures. And you can also see the mitochondria here. So we do not require any labeling, and time to data is the order of seconds. And there's almost no phototoxicity because we do not use any labeling agent, and the use of the light intensity is very low, and there's no energy transfer. And we also provide 3D, even though I only provide the 2D section in this slide, but I will show you another 3D data in the following slides. And resolution go down to 110 nanometer or some for our first generation machine, and 150 nanometer our second generation machine. And the quantification is very powerful because there's a linear relationship between refractive index and biomolecular concentration. And those are main key benefits of using HET in the study of the live cell and analysis. So no labeling and low phototoxicity and long-term fast imaging, high resolution 3D, and quantitative analysis. And let me first talk about the no labeling. Without introducing any labeling agent, we can provide very high contrast imagings of live cell using light. And you can see cell membrane and membrane protrusions, and those are mitochondria and nucleus membrane and nucleoli and lipid droplets are easily measured. And even you can see the heterogeneity in the nuclear plasma as well. And those structures can be easily observed without introducing any complicated and time-consuming the sample preparation step. And it can also overcome the fundamental limitations of the use of the fluorescent proteins. when you use fluorescent proteins, even though they can provide very high molecular specificity, but sometimes you have to worry about cell-to-cell variations and low expression level. In this case, we have fluorescently labeled mitochondrial structure here. But if you look at the cell in the middle, it has the well-expressed the fluorescent signals. But if you look at the cell in adjacent locations, such as the cell outlined with yellow dashed line, in our case, in HCT, you can see the presence of cell and cellular structures and mitochondria, but in this case, they are almost, you can barely see the cell structures or mitochondria because of low expression levels of the specific, the staining agent here. And due to low phototoxicity, you can apply HCT to the study of the neuroscience or the iPSC, where you have to worry about cell viability very carefully. In this case, this is primary cultured the neuron cell, and you can see the cellular networks and dynamics of the neuron cell, and lamellipodia growth cone can be easily observed. And this is like 12 hours movies, and you can see the growth of the lamellipodia cone to the other the cell. So this is one examples to showing the power of the low phototoxicity of the using HCT. The second benefit is the long-term fast imaging. And one example I like to show here is the cancer cell line HAP3B, and you can see the individual subcellular organelles, as I mentioned in the earlier slide. And we have applied the culture medium for in the beginning, and we have injected H2O2 in order to study the cell's behavior in response to the reactive oxygen species. Now you see the dramatic alteration. The mitochondria shape becomes like segmented, and you can also see the formations of vacuole structures. And when you recover to original cell culture medium, you see some of the cells are died, but some other cells are recovering their original shape. The vacuole structures are disappearing, and you can also see the distortions of the mitochondrial structures, and other subcellular organelles are becoming to their native the healthy stage. So you can see these fast-varying changes of subcellular organelles for a long time period. So this is the power of the HCT. So some of our users have used our HCT system for various intriguing studies. In this case, the ETH group has used a microfluidic device to culture the yeast for a long time. In this case, they have measured for 5 days. And now you see very stable measurements of this tomogram, label-free, and the growth of the yeast and the buddings of the budding of the daughter cells can be observed for a long period of time. It's very high resolution, and the very the short period very fast the short acquisition time. And when you say long-term imaging, it's not hours or days. It's like the weeks and months, because cell maintain their original stage very nicely, because we have the incorporated high quality incubator inside of our microscope. You can just keep watching how cells behavior. So this is examples of 3D tomograms of the lead differentiation process of human-derived fibroblast-like cells. On day one, this is mostly like the fibroblast cell. And as time goes on, you are watching the same field of view. And some of the fibroblasts are becoming like the adipocytes, and they are generating the formations of the lipid droplet, which has very highly effective index. And same field of view you're watching onto days of the 42. We stopped the experiments due to other experiments, but if we haven't stopped, you can just keep watching for forever, as long as you can keep providing the nutrition and the other materials. You can keep watching the cell without any limitation. And phototoxicity and photobleaching is another challenges in conventional microscopic technique. But since we do not use any labeling technique, you can just avoid any issues with the photo bleaching. In this case, you're watching the timeless imagings of the cancer cell line, and the top rows are showing the HD data. And second row is we have applied the AI algorithm to retrieve the locations of the lipid droplet. And we have compared the fluorescence using lipid dye, and at when time is equal to 0 minutes, you see the strong fluorescence signals. But if you keep watching over time, the fluorescence signal goes away due to the photo bleaching. So you cannot, this is irreversible processes, as you know, so you cannot watch again the same cell. But in this case of the HET, there's no issues of photo bleaching. So you can just keep watching for a very long period of time without any issues of the low signal to noise ratio. So high resolution 3D provides the unique features of the HT. So now you can see the various subcellular organelles, and you can see the, for example, mitochondrial fission and fusion dynamics. So this is very hard to observe phenomenon if you use conventional fluorescence techniques such as confocal microscope and so on. But when you use HT, you can measure the very fast dynamics for a long period of time. So now it opened the door to the new biological findings, we believe. 3D is one of the most important features that we can provide. So unlike the conventional microscope, we only provide like the boundaries of the cell. So this is iPSC, the patient-driven the iPSC images. In bright field images, you can barely see the internal structures of subcellular organelles, but you can see the outline of the iPSC colony. But if you look at the slice of the reflection index tomogram, and you see the various subcellular structures, if you zoom in the red area here, you can see nucleus membrane and nucleoli and mitochondrial structure here, and those are lipid droplet. And if you focus on this in the middle area, if you use the bright field microscope, you cannot see how many cell layers are present here. But using our optical sectioning by scanning from the actual position, now you can see the various layers are present in this small field of view. So this is the power of the 3D imaging capability. We can measure samples up to the size of 150 micrometer. So embryos, like the human and mouse embryos, can be easily measured without any staining or modification. Those are the mouse embryo at the stage of 2 cells. You can also observe the sperm cells in the adjacent area, and also heterogeneous structures of the nucleus and the two cell stages. Organoids is one of the most fast-growing field, and the size of organoids varies depending on application, from like 50 micrometer to the 1 millimeter size. But we can measure up to 150 micrometer organoid size, and this is what you can do. You don't have to change any protocols. So you can just keep organoids in matrigels, and so organoids very happy and live, and they grow over time. And now we provide high resolution 3D, so you can even observe the subcellular structures and the mitosis, and you can also see that the formations of apoptosis. So those are the information that we can generate. So this is 3 hours of movies, and you can you can also observe the organoids for days and weeks. Okay, the next benefits of using HET is quantitative analysis. Unlike conventional microscopic technique, HET provides very unique quantitative analysis capability. This is based on the fact the refractive index is linearly proportional to the dry mass concentration of biomolecule. So which was first reported by the Dr. Better in the Nature in 1953. But after the development of the HET technique, now we can measure the refractive index tomograms of the cell with high resolution. So you see now we are able to analyze the dry mass concentrations of individual subcellular organelles with very high resolution and very high precision. In this case, this is hepatocyte, cancer cell line, and those are the lipid drumlet. Lipid drumlet has very highly effective index compared to other part of the cytoplasm. So now you can use some segmentation to take out this lipid drumlet, and from this effective index value of the specific individual lipid drumlet, using this the proportional value, which is known as the effective index increment, you can calculate the concentrations of the lipids in each individual lipid drumlet. And HT is also the volumetric 3D imaging capability, so it can also provide the volume information in femtoliter units. Now you have two quantities. One is the concentration, and the other one is the volume. Both are in quantitative units. And if you multiply those two values, you can retrieve the dry mass of the lipid drumlet. You can also perform this to the other subcellular organelles or the bacteria, the bacterial spores, any like the biomaterials, any optically tranced materials, you can apply the similar quantitative analysis. So you have concentration, volume, and mass, and those are the information quantitatively you can calculate from those HT imaging data set. Since we can do this quantitative imaging analysis, let me give you one example here. So this is the same movie that I have shown in the earlier slides. From fibroblast, cells are differentiating into the adipocytes. And using our software, tomanalysis, you can do simple segmentation to rule out the lipid drumlet based on their refractive index values. And then you can perform the calculation such that you can retrieve concentrations of individual lipid as a function of time, and you can also calculate the volume of the individual lipid drumlet. And from those two information, you can also calculate the dry mass of the individual lipid drumlet without introducing labeling agents, and we can also provide various quantitative parameter here. So holotomography does not compromise anything in terms of the live cell imaging. This is very famous diagram in this paper. So saying that sample has spatial resolution, temporal resolution, and signal-to-noise ratio are all important in live cell imaging. But this is very difficult to achieve all of them at the same time because, like for example, in confocal microscope, it has strong, very powerful signal-to-noise ratio. But you have to worry about sample health and temporal resolution. Super resolution technique increases spatial resolution beyond the diffraction limited spot size. But again, the sample health and temporal resolution is limited. Light series is very powerful technique. It improves the temporal resolution significantly compared to confocal microscope, but still they have to use the fluorescent labeling agent. And those are the limitations of light sheet. But in our case, we don't have to worry about the sample health because no labels. And resolution is better than slightly better than confocal microscope. And we also have strong signal-to-noise ratio. And temporal resolution is very, very powerful. So like, for example, we can just measure every like a few seconds, you can measure this tomogram or even faster, depending on the models or the realizations of HT. So due to these various features, there are some leading group has the rapidly adapted the use of the holotomography system in their the discovery of the new biological findings, and there are more than 60 the leading institute and the biotech and pharma company and the university has already adapted the TomoCube's holotomography system, and they have published very interesting papers. So for example, in this case, Princeton University have used the HT to study the biomolecular condensate in the context of the bone metastasis and so on. So those are the few examples how the early users have pushed the limit of the science by introducing the holotomography system in their study of the 2D and 3D biological system. And at this time, let me introduce the features of the second generation holotomography system, HTX1. So HTX1 use LED, not laser. So it provides very high imaging signal to noise ratio, and the spatial resolution go down to 156 nanometer, and actual resolution is 1.07 micrometer. And field of view of the one shot is around like 200 by 150 micrometer. And it has the motorized stage with auto focus module, so you don't have to worry about the focusing at every different positions. And optic lens is 40x, NA of like 0.95, and air emergent, so it's very easy to use. And there's also correlative imaging capabilities provided. So 4 excitation channels, and you can use 3 channels of array. And as you can see here, it also has the on stage, the live cell incubator. So you can just measure the cell over time, because the CO2 temperature, humidity is keep controlled and maintained over time. So there are like several features such as user-friendly interface and practical convenience and the high performance robustness. For example, you don't have to prepare like a special anti-vibration table, because our system is ultra stable. It has the features of enhanced resistance to external vibration. So you can just keep watching by placing our HTX1 system on just rigid table, and you can just keep measuring various intriguing biological studies. And the another feature I like to mention here is that this is calibration free. You don't have to find the background. You can apply to the confluence cell, and you can even measure 6 samples such as the organoids and the multi-layered cells, unlike the first generation HD system. And it also has the 3D, advanced 3D fluorescence imaging capability for correlative imaging features. It also used the deconvolution, so the imaging quality of fluorescence is also good as the conventional microscopic system. Having said about like correlative imaging, we provide high performance 3D fluorescence imaging on top of the HET. So for example, in this case, this is the examples of the organoids. The size of organoids about 100 to 150 micrometer here. And you can measure the 3D images of the refractive index using HET. And if you prepare the organoids fluorescently labeled, and you can measure both 3D fluorescence as well as HET, and you can do correlative imaging. And the right panels, you're watching the correlative imaging to the case of the cancer cell line. So in this case, the overall cell shape are shown with the our HT features. And we have used DALPI to label nucleus, and mitotracker for mitochondria, and lipid for imaging lipid droplet. And we also have the strong the tile scan and stitching features, which are all done in our software. So this is the 4 millimeter by 4 millimeter 3D tomogram of the adipocyte, fully differentiated adipocytes from primary fibroblasts. So you can see individual cells and the well-differentiated the adipocyte, and there they produce the lipid droplet structures can also be seen here, and other subcellular organelles such as nucleus and mitochondria can also be seen here. And like Google Maps, you can just go in the backend first. This shows that we provide the very powerful and excellent tile scan and 3D stitching performance here. And there are several comparable labware. You can use the commercially available multi-well plates up to 24 wall. And as long as you have 1.5H, the bottom thickness, glass or polymer, you can measure, you can use various types of the sample, the comparable labwares, including microfluidic device and microscopic slices, and so on. So regarding the sample preparation, sample conditions, as long as samples are optically transparent and the thickness is around like 150 micrometer, you can measure using HT system. And when we get questions about the sample condition, if your sample meet these conditions, we recommend just do a demo, because there's always we saw the unexpected good result. In this case, this is human hair cuticle. This is not electron micrograph. This is our HD data, but you can see the tomograms of the hair cell with the high resolution visualizations of the hair cuticle structures here. This is what we didn't expect it, but it can be easily observed using our HD X1 system. Okay, now I'd like to move on the quantitative analysis features, so using tomo analysis. The tomo analysis is a software to systematically visualize and analyze the HD data acquired with our HD system. So first feature is the visualization. It blends the two different data sets, HT and 3D fluorescence can be blended for the correlative visualization. So this is a screenshot of our software. And then there are different types of visualization. The first type of visualization is using refractive index values for visualization. So we just changed the spatial distributions or the transparency or coloring based on the RI values. So now you can see the three-dimensional visualizations of neuron cell. And this is also three-dimensional visualizations of the mouse macrophages over time. And different visualization might be helpful depending on the types of research. And in this case, in the right panels, those are the small intestinal organoids, still label-free, and they happily grow in the matrigel without any modification of protocols. But since we have three-dimensional volumetric structures after reconstructions of this imaging set, in order to visualize, you can also color code the depth of the maximum refractive index value and project into 2D for the effective visualization. And this is one example here. So you see different layers and different locations of organoids. And then that can also be used to the monolayer cell. And you can see the even the cell's monolayer on different parts or subcellular structures have different actual position, as can be seen here in these left-hand side images. And sometimes you want to see the three-dimensional visualizations and rotational movies. So we recommend use the maximum intensity projection. But this is pollen images. So you can see the internal structures with like the rotating as you like, and you can just see the you can just view these 3D structures from different viewpoints of your interest. To analyze and to retrieve the quantitative values, we provide the various analysis features. So one basic but important feature is to use the manual segmentation, manual analysis by using manual segmentation. So we provide several features such as polygon and circle and so on. And using this segmentation, you can retrieve the surface areas and the refractive index, the values and concentration and so on. And we can also provide semantic segmentation based on the values of the refractive index. In this case, this is the liver organoids, and using the specific refractive index values, we have performed the semantic segmentation. So you just segmented the whole organoids out of the background, and you can also calculate dry mass and overall mean average concentration and the volume and other parameters such as sphericity and surface areas and so on. But in many cases of studying individual cell, you want to perform the instant segmentation. Like you want to segment the individual cell differently so that you can retrieve different quantitative values for each cell segmented after segmentation. So for this one, we provide the multi-layer the pipeline, the analysis pipeline. So for example, you can use the different values of the refractive index for thresholding to provide seeding of the different locations of the cell in this case. And by adjusting the different refractive index values, you can perform the identifying the individual cells using the initial level 1 thresholding values. And then by adjusting the RI thresholding values by using different levels of the thresholding, you can achieve both the cell instance identification as well as the semantic segment the membrane segmentations of individual cells. So different coloring means the different indexing of cell segmentation, and now you can perform the quantitative imaging analysis for the individual cell. So you can calculate the concentration and dry mass and volume for each cell of your measurement. And sometimes you may also rely on the signals of the fluorescence. So in this case, we have prepared the co-culture, and there are many different types of brain cells. And there's one neuron cell, which is expressed with the GFP. And now we want to retrieve the concentration or mass of the individual cell. And we use the fluorescence as the mask information. And using this fluorescent signals as guidance, we have masked out the HD data of the individual cell out of this whole 3D data set. So this is one other example how you can combine this correlated imaging, and you can also provide, or we can also perform the quantitative analysis based on HT, guided by the fluorescent segmentation. Once you exploit these features, you can do various unexplored study. So for example, in this case, if you use the correlated imaging features of our HD system, you can see overall HD images as well as the multi-channel 3D fluorescence. And using fluorescence as the mask for segmentation, you can perform the segmentations of individual nucleus in each cells, and you can even perform the segmentations of individual nucleoli, and from which you can perform the retrievals of the dry mass and concentration and volume and individual subcellular organelle levels. So we provide both semantic and instance cell segmentation, and they can also be guided by the fluorescent signals as well as the reflectance thresholding. That is very well working for the sparse cells, and that is also working for the confluence cell. And sometimes there are some errors when you're trying to do segmentation. When we also provide the features for editing the cell mask, so you can do fine tuning by providing the feedback to our software. Okay, with that, I have gone through the several the introductions of the HT system. First, I talk about the principle of HT and the benefit of using HT, and how you can use our latest versions of the second generations of HTX1. And I have briefly introduced the features of thermal analysis. So with that, I'd like to thank for your attention and be happy to answer to the question. Thank you.
Thank you, Paul. That was an excellent presentation. We have a few questions from the audience already. If anyone else has a question, please feel free to post this in the questions box that appears on the top right of your screen. But before we start the questions, I think we'll have a look at the results of the polls that came up during the presentation. So first one was, what is the most important feature for live cell imaging? And as you can see, more than half answered low phototoxicity, and 40% said high resolution. And then the other ease of use, there were a few answers. So what do you think of this, Paul? Is that what you expected to see?
Yeah, I think this is like a very interesting result, because we were also think about many responses regarding the imaging depth, but it's good to know that many of our audience are interested in low phototoxicity and high resolution, which is the strong strength of our holotomography system.
Yeah. And then we had another poll. I don't know if we can pull that one up as well. That's asking about people's dream features in microscopy. And there was a more even spread of different features here. But live 3D imaging was the most popular answer. Again, is that something you expected to see?
I was curious about the responses, and we are very pleased to see that the live 3D imaging and label-free organelle observation is feasible now, and automated analysis and quantification of concentration is manually possible previously, and now we are very pleased to launch the tomo analysis in a few weeks, which will enable automated analysis for the quantification and segmentations of organelles. I think that will be very useful for many of the scientific studies.
Yeah, it's responding to a lot of these dream features that people have. So the first question I've got for you is about resolution. The resolution has negative correlation with field of view and capture speed. So what is the maximum limit of resolution in holotomography?
Okay, so the resolution is in principle determined by the wavelength and inversely proportional to the numerical aperture of the object lens. And the principle of holotomography system is the employs concept of synthetic aperture. It's like this structured illumination microscope in fluorescent version. So we can measure down to 150 nanometer, so better than confocal microscope. So that's why you're able to see the subcellular organelles like the mitochondria, feeder, and fusions with very high signal to noise ratio.
Now we got a question that came in very early during the talk asking, can you use fluorescence together with holotomography? And later on you showed that you can. We actually have another question that just came in. Is simultaneous acquisition between holotomography and fluorescence imaging possible? So maybe you can answer that.
Yes. So our HET-X1 system has a features of measuring both HET and the 4 channels of the fluorescence in 3D. So you can perform correlative imaging. So I mentioned about the principle of HET, and for imaging 3D fluorescence, we use a multiple G stack and perform convolution, deconvolution algorithm, so that you can get the images of fluorescence compared with the confocal microscope, and you can overlay 3 channels same time, so we can provide correlative imaging features.
And obviously you showed examples from several cells. We got a question asking, does this work with microalgae?
Oh, yes. Due to the sake of time, I didn't present the result of microalgae, but many of our users have already demonstrated imaging of the microalgae and as well as quantifications of lipids inside of live algae over long period of time. And even C. elegans and as well as tardigrades can be measured without any preparations or fixation or staining. So I think that is also a very exciting story.
Yeah, wow. And can you download the raw data or pre-processed data with the AI system to work with the data in Python?
If I understand the question correctly, I assume it is asking whether we can export our HD data for further AI analysis using the different languages such as Python or C and so on. I think it's possible. Many of our colleagues and collaborators have already demonstrated by using HD data for the advanced analysis using machine learning approaches for imaging quality improvement as well as cell type classification, and virtual staining of fluorescence is also possible. So there's the review article will appear in a few weeks in Nature Methods that will discuss about the use of the HET in combination with the AI.
Great. And this was an interesting question. Well, obviously, we talked a lot about holotomography, but what does the prefix holo actually mean?
I guess that is due to my pronunciation. I guess I saw this question pops up when I was talking about the visualization. So there are several ways of visualizations of HD data, and one, because we have 3D data set, and each box cells or pixels in 3D has the quantitative numbers, and you have to you can decide the way to visualize those 3D information in 2D monitors or 2D the format. One way to visualize is that you can find a maximum value of refractive index along axial position, and then you can color code the G positions with the maximum refractive index value, so that you can color code the maximum the locations of maximum the refractive index intensity, so that you can have pseudo coloring presenting both the overall shape of cells or 3D organoids in terms of their relative location. I think that is related to this question.
Okay, thank you. Thanks. And we've got a few questions about what you can see. So can you see reticuli, Golgi, small trafficking vesicles?
Okay, so I think this is very important and intriguing question. So as we have presented during this webinar, major subcellular organelles such as nucleus and nucleoli and mitochondria or lipid drumlet, they can be easily identified. But there are some structures looks like Leti or Golgi and other structures, but one of the limitations of the HET is the limited molecular specificity. That's why, in order to confirm whether those tiny structures that is visible in our HET data to matching to specific types of subcellular organelles, you may rely on the use of the fluorescence as a correlative imaging capability. But we also working on the development of the AI-based the virtual staining. So we have demonstrated in our recent Nature Cell Biology paper that you can use the GAN networks to predict the locations of fluorescent proteins without introducing fluorescence based on the HT data. So we are also trying to develop further so that it can be combined into our tomo analysis in the future, so that our users can identify the Leti or other subcellular organelles without using fluorescence or any confirmation.
Okay, thanks. And then just jumping ahead a little bit, because there's a related question from Isabella asking if you can see the parts of the cytoskeleton like microtubules or actin.
So you're right. In the low HET tomogram, you cannot see the cytoskeleton because of their width is very, very thin compared to our resolution, and also with those cytoskeleton does not have distinct refractive index value. That's why it is not directly visible in our HT data. But as I said in the previous question, we have demonstrated that by combining HET and AI, we have shown that we can actually predict the presence of the actin or the other cytoskeletons without using staining. The accuracy was about like the 97%. But I have to say that this is not commercialized yet, but we have uploaded the Python code to the web together with the paper. So anyone who has a data measured with TomoCube machine, you can try by yourself.
That's good to know. Thanks. I'm just keeping an eye on the time. Let's see. We've got quite a lot of questions. I don't know if we have time to go through all of them, but there's a few that I definitely want to ask you. This one is interesting. Can you describe the mechanism for changing the imaging or illumination angles? So you showed some different angles, and how do you change them?
So let me put it this way. So we have developed the 2 generations of the holotomography system. First generation is based on the use of laser illumination, and we have used the digital micrometer device to control the illumination angle using the holographic controls. And second generation, which I have mentioned in our webinar, is based on the use of LED, incoherent light source, not laser. We have developed more advanced forms of the light field modulation using digital micrometer device. We have also simplified imaging system, but the second generations of HGT can generate a better imaging quality compared to the X1. But in short, we use the LED illumination together with the DMD control. So we control the illumination wavefront, and we measure the multiple illumination patterns, and we measure corresponding the intensity patterns from which we can reconstruct three-dimensional tomographs. So yeah.
Okay, thanks. I'm jumping ahead a little bit. This is the question from Mohammed. Do you need special preparations for the sample before you test them? So is there any specific sample preparation?
Actually, no. So by putting any samples into our microscope, and as I said, there's no calibration and no, background measurement, not at all. And if you click the button, it takes 3 seconds to measure one field of view, one tomogram. So nothing is required.
That's good. That sounds really easy.
We also provide virtual demo. So if you want to learn further, you can contact info@tomocube.com to discuss and arrange a virtual demonstration.
Great, thanks. We have time to do a few more questions. This is quite a long question, but it's interesting. Is it possible to control the microscope programmatically via a programming language such as Python? This would open up huge possibilities to perform reactive microscopy, as is often reported in journals such as Nature. Is it possible?
Oh, I think this is very interesting question and the way the future direction. So as of now, our team is dedicated to develop the user-friendly software so that you don't have to worry about like complex settings and other adjustment in our software. By clicking on a few buttons, you can just measure and analyze imaging data in one sequence. That's why currently we do not provide such features, but I think that is very interesting. So I'll think about that. But related to that questions, so we have the two ways of the enhancing the capability by users. So first one is we have the C mount, so that the users with the HGT system, they can combine the external module such as confocal module and super resolution module, and they can just measure the various advanced correlative modality. And second one is that, as I said, we can also provide the export features so that you can export our three-dimensional the holotomography data in any form, so the mostly frequently used form, so that you can use ImageJ or the other advanced imaging software by your own code, and you can perform the more advanced analysis.
Thanks. And another question that I saw here is, can we use this imaging system to apply in an organ on a chip platform?
In principle, yes, but I have to say that the working distance of optical lens that we use is about there's some limitation. That's why we only we can measure a sample with up to 150 micrometer. So typical microfluidic device can be used, but like the big platform such as the organoid chip with a huge gap between the bottom layers and the intermediate layer due to the limited working distance, and there's some distortion in imaging quality. But we're also working on the improving the penetration depth so that we can just cover the bigger organoids as well as the organ on a chip. So I think we will have a chance to announce within this year about that development.
Okay, that sounds great. And then the last question I have for you, let me see, let's just, let me pick a last question from here. What about this one? Is it possible to combine tomo analysis with omics or any other system like that?
Okay, that's very interesting questions. I have to say that there are several of our users have already started working on this exciting project. So, like in general, spatial biology is a big field. So many of our users have installed the TomoCube machine in their lab, and they are, as far as I know, they are taking very interesting the research direction by their own. So I think we will see some exciting data from our users.
Yeah, it's looking forward to seeing what people do with it. Well, thank you so much, Paul. That brings us to the end of this webinar. Thank you again for a very illuminating presentation and a great discussion. And thanks to our sponsor, TomoCube. And finally, thanks to you, the audience, for taking the time to attend and listen in. So until next time, good luck in your research, and goodbye from all of us at TomoCube and Bite Size Bio.